This lab was at Urs Gaudi. I think he's I think which place is not Lausanne. Maybe he is a researcher at one of the universities there, so he has access to a lot of equipment. Also does it at home, I guess. Much of the equipment he makes. It's part of the Hecteria network. Last week I told you about these networks. This is a part of a Hecteria project. But we're waiting for the kit to come out, of course, and then we can do it here as well. So yeah, maybe getting back to this canvas, we already discussed this in the past, like which nutrients. It's very important to select the right nutrients or the right atmosphere, pH, and temperature. But now you kind of understand a little bit more why that is, and also, yeah, all these things that we've been discussing are part of the canvas. But you do have to realize that cell growth and production is a very complex process. It's not like just the optimal temperature is enough. The need for certain nutrients changes when you change the temperature, and the optimal atmosphere changes when you change your nutrients. So yeah, you cannot just isolate one parameter and optimize it. And it's all due to the fact that DNA gene expression is influenced by temperature, nutrients, pH. So the process of DNA translation into RNA and RNA translation into proteins, in all these steps and all these feedback loops of thousands of genes that you have in a bacteria or in a human, all these steps are influenced by each other and by the nutrients and all the external parameters.
Can you say that the expression is like communication?
Whether expression is a communication system? Yeah, so there's a constant feedback loop going on. Nutrients, lots of genetic circuits, they control themselves. So the product of the gene, so for example protein, would stop the production of the protein. But then if the protein is then deteriorated or used, and it's gone again, and then it's no longer blocking its own production, and then it's produced again. So there are these automatic feedback loops all the time going on. But yeah, what we really want in a biofactory is a strain or some kind of a species that can have a high yield, so that can convert a substrate, and a substrate is a technical word for a carbon source, for example, sugar, into a product. We want to have an organism that can grow fast, because we don't want to wait all the time. We want to have a strain that produces very low